عليكم ورحمة الله وبركاته. رب اشرح لي صدري ويسر لي أمري. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. Inshallah ta'ala, we'll be getting ready for the 22nd Our Golden Lab. Inshallah, bear with me. The students come in, inshallah ta'ala. Please make sure that you place all your belongings where they need to be. Uh, you begin your pre-lab at 10:10. So if you want to look at your lab manual to just revise and prep for the pre-lab quiz, that's also okay, and it'll help hopefully for your completion of today's lab. And if you need like to look at the lab notebook, I can show you mine. Okay, great. Try to review in the meantime. Remember, trying to remind the folks. Make sure that you wash your hands before. Wash your hands in the meantime. You can check if anyone trying to install. If you're not actually sticking out of the camera, try to push it in so that way it doesn't, hit someone who's walking by. Or we can do that. On your tablets, wait, I will see it, so for those that are wondering, just don't forget to place your belongings accordingly. You know, your hands are possible. And so yes, we will be using drugs today, but you're doing a lot of music for the other ones. You can do it if you can do it after you're done. But won't prevent you to begin. After you take out your mind, you can get it now. You can hang it on. And so we will take 10 minutes time. Make sure once again, folks, this is the attendance, so you have maybe a dozen, and you have the materials for play that you need. If you ever need a calculator or any additional skills, you can write down. question, Mr. Post strong leadership. In terms of like refreshing anyone's questions from the past. So, folks, it seems that we have 16 people, and what I recommend then is, if possible, because we'll likely be groups of four, if Nia and Simone, if the two of you can move to this side, because we'll be working as groups of four, and then separating thereafter, so it'll make things easier. Um, and then I would say for, am I missing one? Five. I say, is there anyone on your left? Anyone who came to sit by you on your left side? No? Okay. Yeah, so if it's okay, actually, Ethan, when you're done, can you sit, I'll tell you, we'll rearrange accordingly. Yes. Okay. So how long do you guys need, maybe just as an update? How many of you have done lab quiz? Can you raise your hand? Okay. For those that need a bit more time, how much time do you need? 3 to 5 minutes? Is that okay? Yeah. 3 to 5 minutes? Okay. So we can wait in the meantime. In the meantime, if you're waiting, you can definitely do a few things. Amongst them, another is, using the lab manual, having a look as to the three different main exercises we'll be focusing on today. One that will carry on, as you could see, for lab six. So that's something you want to bear in mind. Yes. Well, actually, let me see directions. I'll let you know. Just to confirm, is Sina here? Is Sina here? So I got you all from K. What I'm gonna ask is that let me just shut this one. As you come in, try to do the lab quiz as soon as you can. Don't mind like the grades. Um, if anyone hasn't been marked present, just let me know. But I believe we have 17 of us today, which is great. Um, but I just want to make sure in about 2-ish minutes, 2 to 3 minutes, we'll begin. Who's excited for today's practice? This lab is going to be interesting. You're dealing with isopods, yes, that's amazing. And plants. You get to deal with two different organisms, hands-on, unlike what you did in the zoo. In the zoo, you got to take photos, of course, and you got to maybe see them up close, but hopefully you'll appreciate the different organisms you get to interact with as well today. And something to point out actually is I noticed we've been connecting the dots a bit with different practicals, and so I hope with our last one coming up soon that you can appreciate, the diversity of unique species from inside to outside our lab, from animals to invertebrate to fishes in the sea to plants that we've dealt with, winter wheat, and today we'll be dealing with lemma as well. So hopefully you'll appreciate the different organisms and their different morphological, physiological, and I guess and thermoregulative properties or behavioral responses too. Okay, so we're gonna get started. Hopefully, for those that are almost done the lab quiz, they hopefully are done in this moment in time. Um, but if you're not, just let me know, because I want to make sure we're all paying attention. Bear in mind, folks, as well, you need to let me know if you're using your phone, because I am able to take note of this, so just bear that in mind. So we've just that I went through our pre-lab quiz. Pre-lab quiz number 5, right? Isopods and their behavioral responses. We're going to go through the introduction, announcements, attendance we've just done. So we're going to go briefly straight ahead now until or up to the introduction. And then we'll go actually adjust the setup, going straight to exercise 5, our last exercise, which is essentially involved in the middle bench here. And then for exercise 2, you're just going to be visualizing the isopods under the dissecting microscope. And then exercise 3 is where you're dealing with the different wells that you see and the many more isopods available on your bench tops. And thereafter, we'll have cleanup. So hopefully, we'll try to do this in efficient timing. Any questions, by the way, regarding the outline? So let's just go for those that want to get a reference. Go to page 74 of your lab manual. You'll be able to see where we're getting straight ahead to. First off, so So just like how we've done it before in terms of assays, or in this case, we'll be dealing with the lemnas as a aquatic species, like a plant that inshallah will grow with unique traits. And interestingly, we're going to be doing like what we've done before with the winter wheat, if you recall, of treating with different conditions. But in this case, each group will be setting up 3 different replicates and perhaps it will be better to have you guys oriented in your groups, 3 to 4. Okay. So I'm gonna have um, we're gonna have one member join their group, 2 members join this group, I believe. Let's see. And then, okay, so we'll have like a group of five, and that's okay. Um, so this is what I'll say or suggest. Uh, Nia and Olafunke, can you come and join this group? And then can you join the group down there? Sorry. Actually, I'm gonna adjust it so that way I realize some of us may essentially have some observances wherein they prefer like not to intermix. So what I'll do is the following. Instead, let me just calculate a few things. I don't want to split Jai and Robert, but let's see. We may have to do something. Um, Owen, are you comfortable moving? Yeah? Okay. You're a good man. You're a good man. So what we'll do, Owen, you'll join Jeffrey. You'll join Jeffrey. And I'm just gonna see. Okay, the two of you, Nia and Simone, you two can join this group. And then that makes four of you here, I believe. Five of you. You guys stay as is. Um, Let me just see. I don't want to make you guys uncomfortable. And I want to make sure you guys essentially don't feel discouraged from being separated from your former groups. I want this to be efficient, that's why. But also, if you actually feel that maybe I didn't do justice, let me know. And I hope, let's see. Okay. Um, Okay, so let's get straight ahead. This is going to be important just to help prepare ourselves for the upcoming practicals. As an FYI, if you know folks of our lab, because there's about six of them that are absent today, that you know, for example, maybe are absent for a specific reason, please let me know. Let them know to email me. That way, hopefully we can allow for better coordination of our groups. And that way, for our final practical, you guys can be in your original groups as well. But this is aimed to essentially make things easier for not just exercise five, but exercises two and three. So I really hope, inshallah, you enjoyed the remainder of today's practical. Briefly, going straight ahead, measuring population growth in Lemna. So you're not going to be able to do this now, but you're going to be able to set up for preparing for the final lab, just like how we did with the winter weeks. And so, first off, we look back once again, page 74. You have actually on page 76 detailed visuals for the different groups that hopefully you'll all be taking into account. Let's see. I want to clarify this. Different groups. Okay. Yes, so you guys will be actually making three different reps, but the issue is we only have four groups today. Hmm. So let's see. Likely it'll be the case that two groups will be doing more. Um, let's see, based on numbers. Likely this group will be doing two. You guys will be doing two as well. Javita, if you want to move as well, that's okay to the side. Um, if you feel comfortable, or if you want to switch accordingly. But you guys will be doing two different controls or two different conditions. If you guys look at page 76, you'll see what I'm referring to. And let's see, quantity, 444. Okay, how about I just go back to what we were in the past? Were you guys group one or group three in the past? Trying to remember. You guys were group one, one, two, three, four, five, six. Okay, so there's no group one and two here, but this is what we'll do. You guys will be doing control and experimental treatment one. So we'll be essentially modifying the different concentrations of sodium chloride as a marker for the influx and the uptake of essentially table salt, or in this case, road salt, for the effects they have on lemna growth. Think of it like the winter wheat, but essentially I want to give you guys the conditions now. So you'll be doing the control and experimental treatment one. And then this group from the top right, from my side, or the bottom left, will be doing experimental treatments 2 and 3. And then this group will be doing experimental treatment 4, experimental treatment 5. That way, those that are like also comfortable with the previous groups, they'll be able to hopefully coordinate this. But if you guys want to do more, let me know. But briefly, you guys have now the conditions, right? It's probably going to be shown up in future slides, as you can see right now. But I want you to do the following. How many of you recall, actually, I hope this formula, yes, the formula is here. Page 75. Have a look at that formula because that's what you're going to do to essentially take up the right amount of volume of your treatment within the graduated cylinder. There's two different types, and for the Lemna, one for one for the artificial pond water, which is shown in the painted glass jar, and the smaller one is for your stock. So I just want to essentially look at pages 75 and 6 to denote the calculations of what you'll be doing today. And so let me see if I can give you an example. Um, Let me do an example with, okay, I'll just see if there's any extra info. See that you'll need to calculate V1. Okay, so if you look at the formula, it's C1 times V1. So the concentration of your stock multiplied by the volume of your stock should equal as a ratio to the concentration of your final volume, including the artificial pond water, multiplied by the volume itself. So you're calculating essentially the ratio, and you're trying to determine V1. And so let's just do this to make it easier. C1 times V1 equals C2 times V2. This refers to your stock, i.e. the clear glass jars. And these two refer to your final concentration, i.e. your stock plus the pond water. Don't worry about essentially it means your final solution because the idea is that you're starting off, right, with, let's say your stock. We'll do an example. Concentration for experimental treatment 2 is 1.25 grams per liter, right? The point is that you have a ratio here, and the idea is that you want to isolate V1, meaning you're going to put C1 underneath, and that's C2 times V2 divided by C1. Now you know C1 is equal to 1.25. Who can give me the other points? What's V2? Anyone know what V2 is? Don't you? Microliters, liters, what's the unit? Amount? Milliliters, correct? So bear in mind, you have to actually convert the units. So it means 0.2 liters, right? So capital lowercase, okay? Reason being is because you notice that the units here are in grams per liter, not in milliliters. So you have to thereafter convert, therefore. So that's 0.2 liters. And then can someone give me the concentration, the final concentration of the experimental treatment to condition? 1.25. So essentially, and this may make it easier for you guys. Wait a sec. Yes. Sorry. Let me just make an update. So the stock is at 12.5. But very similar to what we had before. 12.5. And then your aim is, as you saw at the top, is getting 1.25 grams per liter. So if you just essentially do this, you don't even need a calculator because it's very simple. You may notice that the ratio of your stock compared to, or actually let's do the calculation. It's going to be, yeah. Well, 5,000 grams. Oh, really? No, so okay, that's a good point. Essentially, both work. Micrograms divided by milliliters is the same as grams divided by liters. Because it's essentially 1 gram is 1,000 milligrams, 1 liter is 1,000 milliliters, 1,000 divided by 1,000 is one. So essentially, units are the same, they're equal. But the thing, does that answer your question? Is there anything else you found problematic? So the idea is that if you do this, you would essentially get, remember this is in liters, right? So just bear that in mind. You would get essentially 0.02 liters, which in our case is 20 milliliters. I want you guys, so I hope that be done as an experimental example, but I want you guys to do the same for column 3 across the remaining other rows. So do that, repeat those steps that I just showed you, and hopefully you should be able to do this easily in like the next 3 minutes. If you guys are having problems, let me know. And you don't need a calculator because you'll notice a trend, right? Let me know if you guys are having problems. Try to do this next 3 minutes. If you haven't, if you're done, let me know accordingly. So I just passed through a few slides, but you guys don't worry, you'll just be able to do what I just said. And you don't, once again, actually, when you guys are done in about a minute or so, let me know, 1 to 2 minutes, and then we can go over this. So that way, every group will be consistent, and this will be a great way for you guys to also think about the importance of replication and consistency for upcoming exercises, as well as to refresh yourselves upon the FLR, because it's very important that there's certain results, or at least for your results from today, that you'll see next week, inshallah, that you want to make things consistent throughout the course of the practical. Um, so, any questions, by the way, on how to do that calculation across the other columns? Is it relatively straightforward, or are you guys having difficulty? Is it okay? I'm gonna ask for you guys, what was the for those that have calculated it thus far, what's V1 for experimental treatment one? Does anyone know? 2.01? For the first treatment, experimental treatment one. What do you get as V1 in the column 3? Not one, not 0.01 as well. It's 20 mils. So it's actually 20 mils. Let me actually let me ask, who else is struggling? Like, are you guys getting values like 10 mils, 100 mils? What values are you guys getting? So that's correct. So 0.02 liters, just bear in mind the units, 0.02 liters or 20 mils. It's okay. Let me clarify. So remember, actually, if you look, yeah, believe it or not, all of the different relationships pretty much between these two concentrations are different by 10 times, right? So your stock, for example, could be 12.5, it could be 25. Let's say it's 25. And then the final concentration is 2.5. Let's just say as an example. And the same is done for the rest of them, believe it or not, right? So like you'll see that for treatments 4 to 5. And the idea is that if you are starting off, right, if I use this so you guys can understand, if we start off with 25 grams per liter and reach at the final volume or at the final with the final volume of solution 2.5 grams per liter, folks, let's pay attention. If you're going down by 10 times in concentration, then you would technically expect to go 10 times up or higher in volume because there's an inverse relationship in that, okay, if you, let's say, go down in concentration, let's say 10 times, then you're going up essentially 10 times in volume. So like let's say like you wanted to actually does that make sense? The relationship? Does that make sense? I could give an example, but it is a bit straightforward. If let's say you start off okay, so we'll do it with an example. Um, let's see. So okay, if for example you have let's say you're at 25. This is let's say 1 meter. Actually, it should be larger. Scale accordingly. Um, let's say you're dealing with a flask and I'm just trying to see if that's related to that. Yeah. Um, so let's say this is at 1 liter, and let's say this is 10 meters. If you're here at 25, like this is your pure stock, right? So imagine that's a bunch of salt added in. And it's just here at 1 liter. So this is 25 grams table salt, NaCl, right? Does that make sense, folks? Based on understanding this thus far. Now, if you essentially add in a bunch of water, in our case it's artificial pond water, and you go up all the way to the top until the final volume, which is 10 liters, then essentially you're dividing that by 10, the total concentration. And that's why it's essentially 2.5 grams per liter as a total concentration. Final concentration. Does that make sense? Yeah? Are you guys having any problems with this? No? Okay. So bear that in mind, essentially, just like how essentially concentration changes, so does volume. And so as you move up in one way, you decrease in another, right? Because the more volume you have, the less concentrated your solution. So with that in mind, all of them, by the way, should be consistent, meaning that if you look at the remaining experimental treatments, what other final volumes or what initial volumes do you start off with from your stock? 20? For experimental treatment 4, what do you guys get? 20? Experimental treatment 5, what do you guys get? 20? 20? Let me know. I want to hear voices because I want to make sure you guys understand this. Especially like if you guys take chem courses, it's very important. Yes. What do you get? 20? So it's 20 mils all around the board. Okay, so just bear that in mind. V1, you put in 20 mils, 20 mils, 20 mils. But here's the catch, folks. For the third or the fifth column, where you're talking about artificial pond volume, that is essentially the difference between your initial and your final volume, meaning, as in this case, what would be the difference? Anyone just share it out. What's the difference between these two? 9, right? 9 mils. So what you want to do is then for your table is look at the final volume that you're going to reach, which is 200 mils. Subtract that from the initial volume, which you guys know as well. So based on the fact that you see all of consistently the volumes for both, what should the essentially value be for the amount of volume you add for the pond water from the pond water? Can anyone share it out? 180. What other values do you guys get? Anyone get 180? Raise your hand. 180, 180. Okay, what other values are you guys getting? Are you guys maybe, is it difficult to understand for those that are like maybe confused? So I will just say that actually I want to hear you guys because I'm worried. Majority of you are silent, and it could be that you guys are tired, but I want to make sure. Are you guys confused? Elisha, are you confused? No? Owen, are you confused? No? Wasi, are you confused? No? Okay, so let me ask this. Actually, I'll ask Dorlan. What volumes do you need for the pond water? Okay, 20 for the pond water. Pond water. That'll be for the initial. Yes, that'll be for the pond water. Okay. Not 18 meters. Huh? But if you look, okay, so let me show you guys. If you look at column 6, if I'm not mistaken, it says 200 mils, right? We know that the initial for across column 3 is 20 mils for every tree, including the control. Actually, control is 0, so it's already 200. But the idea is that consistently you'll be adding in thereafter 120 mils, 180 mils. So that's correct. I just want to make sure you guys understand this because you guys are going to be doing this as groups, and it's very important that you put in the right volumes. Otherwise, this could be a great concern for your results for your final practical, right? So you don't want to mess it up, hopefully. So just bear that in mind. You can fill it in accordingly, but I just now, in the meantime, want us to go straight ahead and pretty much come to the front with the volumes that I've told you thus far. So control and treatment 1 will be this group here. You guys can split up if you want. Treatment 2 and 3 will be your group here. Treatment 4 and then treatment 5. You guys can see the volumes and be able to compare where they're located. The last one is, sorry, group 4. The treatment 4 is here. 5 is there. I think this is the control, and then the other from the other side. Any questions? What's control? You guys are treatment 2 and 3. Yeah. And I just want to highlight this. If you look at the slide here, you guys are doing 3 reps. So what that means is that you guys should, yeah, so you guys should essentially pick out for each treatment 3 cups and label them on the body, not on the lid, because the lids are going to be used soon or like reused. And so once you're done putting in the right volumes, following up with the procedure from the manual, you guys, when you're done, let me know. Label them with the details that you see there, but also, yes, as shown, the practical, the essentially concentration, and the replicate. So that way we can orient ourselves next week or 2 weeks from now and be able to know the specific conditions that we need. Any final questions, folks? Questions? Any questions? Okay, if not, you guys can get straight ahead. And I want you to try to finish this in the next 10-ish minutes because it's, I'll be honest with you, you guys can do it. And I can trust you guys, hopefully, to do this. Just follow up with what I've said. Remember, V1 is 20 mils, and the artificial pond water is 180 mils. Make sure as well, when you're dealing with the stocks, try to be a bit more closer to the bench top in case, yeah, cylinder, try to move it a bit further back, just a bit further back. So that way you guys don't spill it. You can label them in the meantime. All of the reps should have the same conditions. Perfect. Any questions, folks? Groups that are doing more than one treatment, you guys can use both sides with the extra cups accordingly. But I just want to bear in mind that you guys split to make it more efficient. So, like for example, this group has three and two individuals. I could split into two different treatments. The group at the back can split into two, and then the remainder can either help other groups or essentially help their group accordingly. Complete the other reps. Once your group is done, please let me know. And if you guys have concerns, we'll talk about them in due time, but make sure that you put them correctly. If you have difficulty measuring, place the glass cylinder on the bench top, or have someone hold the cylinder as you pour it in, and hopefully you should be able to do it accurately. For the artificial pond water, use the larger cylinders because it could go up to the amount that you need. Um, yes. Uh, you're doing 180, remember. Oh, for the control? You're not doing the control, right? Two and three. So you'll do 180 with the large cylinder in each of the test tubes. I would say, sorry, in each of the plastic cups. Just bear in mind, start off with the stock. It will make it easier for diluting appropriately. 20 mils in a smaller cylinder. Uh, Put it actually into the cups, each of the three cups. Yes, yes. They're just replicates that you will separate by an individual line or number. You don't have to. If you guys want to put a group number, I would recommend you taking note of your group number or letter in your lab manual, so that way 2 weeks from now you'll be able to know the results accordingly. When you're reaching close to 180, slow down and you could be able to do it more easily rather than shooting up and then having to return it back. Please. That's okay. That's okay. It's okay. Like if it's off, let me know if you guys are having difficulty. But you guys can move further ahead. Have someone hold it for you. Someone should hold the glass cylinder, so that way it doesn't fall. See? You guys are the control, right? I believe Bruce is down here. Yeah. Yes. I just want to ask, Wasi, which group are you? You guys are treatment three, right? You're doing three? They're doing two. Okay, I see. Technically, the control, if I'm not mistaken, is somewhere here, right? Likely where you guys are. So if you can make space for Giant and Owen, or Robert, sorry, to make their way, because they. Let me know how it goes. You can do it. When you guys are done, let me know because the next step is adding in the Lemna. So when your group is done, your job or your next step is to essentially take out four little Lemna plants, healthy ones indeed, and they have to have like three different miniature petals. They're called dahlias. So try to look, see if they have like three different miniature petals, and essentially take out four of them to place into each of the containers or each into the plastic cups. And I would recommend, I don't mind, bring it a bit closer. Or I mean like closer to, because they will have to pick it up. That's for us. No, it's not. Folks, are you guys looking at the stock? This is your stock. So you guys should be here. Has anyone been using the wrong stock? Because that's important to note. If you guys are using the wrong stock, make a note of that so that way no one's repeating the same stock with the same set of reps. Control. When you guys are done, let me know. I'm not sure as well as you guys are done. Is that just getting up or? Wow, that's pretty. That's pretty. So we do all where actually I would be now perfect. So once you, by the way, you're adding in four helpful lemons into each of, lemons into each of the cups. They should have three miniature petals called thalia, so bear that in mind, folks. If you don't, then take it out accordingly and swap it for another. And then once you've done that step, you guys are going to actually close it up with the lids that are available in the middle tray. Once you've done it, let me know. When you guys, yeah. Um, you guys are done? Perfect. Give me just one minute. Um, so, um, when your group is done, remember they need to have 3 miniature petals, dahlias. Four of those lemnas need to be in each cup. Is that the case for you guys? 3 miniature petals and four in each. All healthy. Yep. Or like four plants that have three. It's okay if they're small, but as like if they have the right features, that's good. When you guys are done, you have to bring your cup.
I have a question. Do you think this fit the criteria of having three songs each?
This is the bit where I feel like we were king.
Can we replace it?
It's better maybe to replace it. But if you guys don't, then maybe it's just like part of the panel. Talia eventually underneath.
Yeah.
When you guys are done,
Yeah. That's perfect.
I just wanted to make sure if there's anything else you guys need. So they need to be paid for this. Just make sure you guys do this. So when you're done, remember 4 healthy lemnas with 3 petals each of dahlia. When you're done, we bring them actually here, just like how we did with our plant. I recommend having them aligned for each treatment.
I saw it, I was like, yo, I didn't. I saw it.
Well, if you can also put the treatment number, that would make it easier because we want to align them as the treatment. So that we'll have treatment 4, 5, 6 years, and then 4.
Yeah, that's why. Can we just write them on? Did you not find them?
How long do other groups need before they're done their By the way, folks, please make sure that your lids are on tightly, so that way they don't run the risk of seeping through the paper.
Yeah, yeah. Okay. Take one. Oh.
Don't forget that this need to be applied. But we So folks, start looking back for exercise 2. You'll be dealing with an dissecting microscope. So someone should put it on their at their bench, and then you're going to start opening up the isopod. Specifically, we're dealing with the bills, the ones that we're dealing with today. So they're a bit small, but as you can see, they're dark and they have a unique morphology. We could talk about the water. So, yes. So you're taking out one style bug, so style bug looks again, and it's essentially to clarify, you're just taking out one to view under the microscope.
Oh, just one.
No, we're not cutting it. No dissection. I know the dissecting microscope, but you're not visualizing it. Very question, actually, because a note that I was told in both is that you do not kill or harm any of these cells, so please don't harm them. That's another thing. Sorry, that's fine. Please be careful. Place them on the petri dishes. You guys should have petri dishes, I hope. Yeah, actually, they're already here. Perfect. So you don't have to even take them out. Just visualize them under the dissecting microscope, and just bear in mind that you obviously don't obscure or fiddle around with the control bugs. So that way you can take appropriate annotations of them with the detailed figure captions in the manual with the details that have been described as well. Thank you for game groups. If you guys find it difficult to see that each bench actually has a see this. Yeah, that's another thing. So technically, these bugs are, they're generally speaking nocturnal, meaning that they sleep generally, or they're more tired and fatigued in the day and more active in the night. If you see it in your manual, you notice there's specific conditions that they're most optimal in. Yes. So not for yet. For exercise 3, yes, but we're at exercise 2 right now. And so that's where I just want you guys to start on the dissecting microscope, turn on the lights from the top right-hand side, settings you'll be able to adjust. And you already have the Petri dishes with the isopod, so you don't have to take it out of the vial. I just want to make sure, or of the Petri dish, you guys just need to make sure that you're taking appropriate annotations of them. And in terms of, once again, reiterating, well, actually, this is to come soon. So let's see. Okay, so let's just go all the way back. Does everyone have so actually, let's go back to figure. We have a figure for this. We'll go to your page in your lab manual, page 66. You want to be able to draw the features that you see from your, arthropod, the sow bug. You want to be able to draw the following structures. You can see them as well: the abdomen, antennae, compound eye, head, legs, telson, thorax, uropod. So these are different features that you may already see based on what you found in the manual in page 62 as well, but also from the pages that you can also compare accordingly with. I just want you to be able to draw this in the next 5 minutes or less, and that way we can go straight ahead to the final exercise, final main exercise, and that is exercise 3 for today. Keep it up, you guys. If you guys are having any difficulty throughout the lab, let me know accordingly. Yes, you guys can take photos. I would just say try to draw accordingly, whatever you can, because importantly, you want to make these annotations from your eyes directly with the ocular microscope. So try to essentially view it accordingly or from the ocular lens without like essentially redoing it later on today. I'm just gonna go forward though. So you probably recall from, I think, lab 2, where we talked about the scientific method, and really what I touched upon from an approach I find holistic called the I5 approach. And one of the tenets or one of the components revolves around investigation. So I highlighted this in a past part of my experience in like research, academia, like even around the world, if you like look at the Toronto Zoo, for example, you may have seen like 20 different types of species across many pavilions and different types of features, morphological, behavioral, etc. And really trying to, appreciate and investigate, as we talked about the Solomon Island frogs, remember? We saw, for example, some of them are basking underneath the ceramic heat lamp, and others are essentially, cooling down under the shade in their small pavilions. So you want to think then, like, how can you investigate, create research questions that you can appropriately analyze? I talked about them at Lab 4, but hopefully what you can see and appreciate from this slide is that it's very applicable, having the right framework and right intentions, of course, as well. Because for me, I explore these with a mindset that, okay, how can I best please Allah? How can I best think how these methods could actually be a benefit to humanity? And these things I want you to think critically because I'll be honest with you, from a scientific lens, many things that you will see are not only um, sometimes pessimistic, but oftentimes toxic. So you guys want to think better in a way that like you can see yourself down the road 20, 30, 50 years from now, continuing your profession, understanding investigation, the components of how to be an amazing explorer, researcher, polymath, scholar, etc. in your field or discipline. I just want you guys to really appreciate that because you can see this from a slide and then just pass through another set of slides and just forget all of what we just talked about. But you really need to be able to apply these concepts and actually, from a more authentic approach, see how you can find meaning beyond these slides themselves and beyond the words that I've just described. So think about what I've talked about. Think about what you've done from your FLR. You're observing, you're making questions, you're creating hypotheses, and you're making predictions accordingly. You have from your hypotheses your alternative and your null, have HA and H naught or HO, and essentially both have relationships between your independent and dependent variables. For your independent, which our case, actually let me ask, what do you guys think after you guys have drawn out your arthropod? For exercise 3, what do you guys think we're trying to investigate? For those that have also looked at the manual beforehand for the pre-doc. Yes, so, and really that relates to a concept known as taxis, and essentially a level of attraction of these arthropods to different different environmental conditions. And so your job, folks, is to really analyze these different conditions, but to go back to the point of your hypotheses, HA is going to be essentially you determining or you investigating slash concerning if um, arthropods like the sowbugs, and there's a specific name. I just want to make sure I get it right. Like their genus, the Porcelio subspecies. They essentially, you want to be able to look based on your hypotheses if they are attracted or repelled from a certain environmental condition. If if you're looking at your null hypothesis, you would assume that there is no significant difference, i.e., this independent variable, i.e., the conditions that you place them in, should be irrelevant to the number of sow bugs that are present in each condition. So essentially, like your control, you would find sow bugs equally distributed across all conditions. Whereas for your experimental slash alternative hypothesis, if it's correct, you expect that there would be a distinction a certain number or a more skewed or essentially more favorable selection or quantity of cell bugs in a specific region or a number of conditions, but not on all of them. So, i.e., it's unequally distributed. Does that make sense, by the way? Yes, good. Cell bugs are like those arthropods, the ones that you're looking at right now, mini insects. Sorry? They're not making more of themselves, so you guys have hopefully an extra vial that has 10 of them in. So you guys will actually see, yeah, that's the one that you're looking at, Owen. So essentially, you have one that's in a Petri dish for exercise 2 to analyze morphologically. Exercise 3, you're going to actually be taking them out. And we'll talk about that in about 5 minutes. But does that make sense in terms of like the hypothesis and then really conducting the experiment, which you're about to do right now? The experiment essentially is you essentially looking around. So there are different conditions across the room. Some are on food substrates, like some of them are on chemicals, and others that are essentially additional biomolecules. So essentially, like we have here, food substrates, we have sugars and then chemicals. Likely the same on this side. So because we have a good number of people here, if you guys as groups want to explore a certain condition, then you guys will have 2 separate plates, one for control, one that will be experimental as well. If you guys find one more activity that you want to investigate, i.e. how these cell bugs react to different conditions, like if you wanted to look at the chemicals or sugars or food substrates, then essentially what you can do is move to another location across the lab. So that way you'll be able to look at the different conditions and just focus on, keeping the bucket as it is, but moving if you want to, let's say, explore the hypothesis on how do these insects move or react to the presence of, let's say, food, right? Or to chemicals, chemicals of fat, or to sugars in the middle. And the same would be on the other side. So you guys can move accordingly as the groups that we've designated for stay. So I'm combining, like I'm talking about exercise 2 and 3. Exercise 2, you're only looking at one, the one that's already in the dish from the dissecting microscope. For exercise 3, which is what I'm talking about right now, that's where you're actually going to be taking them out and placing them accordingly. I just wanted to go over this as you guys essentially complete other tasks today, so that way we don't lose track of time. Any other questions? So let me just briefly ask then, how have you guys, like, are you guys drawing the isopods? Are they okay? Have you already drawn them out? Yeah, morphologically, you have the different, like, the criterion, the Europod, the, like, all, I think, seven different features. If you guys have them all, then we can move further ahead. But I want to make sure you guys actually drew them. So please do so if you haven't done so already. I'll wait another 2 minutes. Please, folks, bear in mind that you guys are responsible, right, to take on these tasks. Try to draw out what you see from the dissecting microscope itself. Don't just draw it out of hand. Draw it out, look under the microscope, see the different features, and annotate them. I.e., if this is your arthropod or the isopod, let's say it looks like something like this. And it has, by the way, external gills, which is unique, right? Because it's not like it's an exceptional organism. And they'll have some different features, but hopefully you guys can appreciate the summary of it here. You guys want to be able to thus, like once you've drawn it from the microscope, like with the field of view that you can See, you want to be able to annotate like these are the antennae, right? The straight line is what I was told when I was, you know, a student. But essentially, you draw the antennae or antennae, and then the other features that we talk about, the Europod, etc. And make sure that, folks, you have a detailed figure caption. What I mean is, let's say this is your figure. Say figure 1. Visualization of soy bugs. Put the scientific notation as well, right? In italics, Porcelio, SPP. If it's difficult to write in cursive or in italics, you can also put an underline. But the point is that you include more data, including they use a dissecting microscope, right? Even down the road in other labs, you may include the magnification, and then also highlight in brackets that included with annotations for morphological traits. And that way you can be able to not only make detailed figure captions in the future, but also excel for future courses and your performances therein. I'm just going to briefly go further ahead. Now that, let me just get a show of hands. How many of you have done exercise 2? Raise your hand. I want to make sure you guys actually saw the soy bugs, because this is pretty much your last chance or time to do so. All of you guys, group, I'm going to say your names, Jaden, Wasi, Biha, I got your name. Let me just think. Let me think. S is coming to my mind, but I don't want to make a mistake. Alamina, am I right? Alamina? So, you guys are done? All of you guys? Perfect. You guys are all done? Just to confirm. I just said your names. I didn't ask if you guys are done. You guys are done? No? Give me a minute. Or can you guys be done in a minute or so? Perfect. Okay. So folks, I want you to be able to see these organisms before they just run around, right? So just bear that in mind. Your job though, for exercise 3 is to come up with, a question and observe the soy bugs as they move about. And based on the hypothesis, as I just talked about, their reaction to different conditions or different substrates per condition is something that you want to highlight for your null and alternative / research hypothesis. And essentially, you'll be having a control plate. So you have a tray here to make things easier. You have a tray. Folks, please pay attention. I see some of you on your phone. Please bear that in mind that you all pay attention. So remember, aren't overburdened accordingly. You can have this as your control. You can have the other side as your experimental. And the idea is that I just want to wait till Jaden's done. You're done, Jaden? Okay, let me just go through the slides. It's okay, keep going. I'll talk about the wells afterwards. Actually, it's okay. Let's go forward now. You guys have a look at your bench. There's many different materials available. If you look here, inside the paper towel, you'll have inside 2 different halves of essentially this filter paper. One will be for your experimental tray with 5 different wells, and another will be for your control tray. And your goal is to do something quite simple to start off, and that is to open up the tray. If you guys want to come on this side as well as in the back, or actually those that are in the back, I want you to open up yours so that way you can actually do real time. Open it up. Everyone can open up theirs, and that'll make it easier. Open up your treats. Open up your treats, everyone. And I want you to do the following. Each group, once again, has a paper towel with 5 different sets of filter paper, right? So you just add the filter paper into all 5 wells, including the middle. The middle is where you're gonna have that last line that you just covered. That's where your sowbugs are gonna go in. But before they go in, what you need to do is the following. achievements for some. Start off, folks. Let's pay attention. Sorry, I know you perhaps doing it in real time. Did you add in the filter paper? Perfect. So what you're gonna do is add in water. Essentially, add it in, not like over blotting, but add in a good amount on the edges, drop by drop, until essentially the water, sorry, the filter paper gets mildly soaked up. So don't overdo it, because you don't want residual, like excess water spilling out. You just want to add in a fine amount for each of the wells. And you guys should see it here. So each group should be able to accordingly add in the water. And then, sorry, before I should stop, I forgot, there's an important point. Pause for just a moment, folks. There's an important point for this assay. For those that are doing it for the experimental, only do the middle. Remember, you have two different trays. One is your control, one is your experimental. The control tray, which should have all 5 filter papers in each well, will have only water all across, right? Because you want to see the different conditions. And so what you need to do, thus, for the experimental, is only have the middle well, only the middle with water. The other ones, now it's okay if you guys want to change it out, because you can move accordingly in your wells themselves. But I want you to bear in mind that, let's say, this group wants to look at the effects of soy bugs or the effect of food on soy bug responses or motility, then for behavior. So what you want to do is then essentially move your experimental tray or your both of your trays to that specific station. Don't move the bucket itself. You guys have an opportunity to essentially go to different categories and just focus on that one that you want. And we should be able to move accordingly. But I just want you guys to bear in mind that the group is already here. Try to see another experimental treatment condition. Like your find food is already taken from both sides. Look at the sugars or the chemicals. And that way you'll be able to see the effects of their responses in real time across the different fields. Does that make sense, by the way? Anyone have any questions regarding your research question, your hypothesis, or even what you're doing today? Any questions for us? So by the way, I'm just going to finish this, and then bear in mind that some of them may appear damp. Try not to do it too much. I'm just in a bit of a rush, but I want to get take your time, do one drop at a time, let it get soaked up, and then it should essentially become moist, or at least have water present on the filter paper. Now, what's next up is that you are, okay, if you're, let's say, in an advanced biology course, you actually shape chemicals up, or even bacteria up. And I say this because essentially you may have them not be suspended well, and you would have had maybe your chemicals or your materials just at the very bottom. So generally speaking, I recommend just mix them just a bit, and then your job is based on the different locations. Starch, okay. You'll see the pipettes having labels. So do what I just did with water, but place them into the correct wells. For example, you'll have the wells that are oriented by number, but also with ingredients or with substrate. So this is for starch, this specific pipette, to make sure you use the right pipette accordingly. And remember, don't overdo the drops into the filter paper. Just do it drop by drop around this exterior, and then slowly towards the center until the entire filter paper is covered with your substrate and prep. Don't overdo it because thereafter may obscure. You don't want to essentially create errors in your results. So that's what I want to say. Are there any questions, folks? We move further. Yes. But the controls, for example, have all of them water. You really just want to see controlling for any substrates. How do these sowbugs move? If they move, ideally they'll move in equal distribution randomly. But let's before I move further, you guys are going to be able to create predictions in advance in real time. That's a way of building up a level of acuity of knowledge and awareness of how you'll be able to go forward with these practical. When you are done adding in the wells with plates, this is with the filter paper. I want you to do the most interactive part of the class, and that is taking out these sound bugs. Folks, these are living organisms. Bear that in mind. Don't get too grossed out. They're organisms like yourself. But the thing is, what you're gonna do is the following. I'm gonna show it to you guys. I want to actually do it. So I'll do this stuff for actually it should be okay. Let's see. There's two main ways of doing this, and so I just want you guys to pay attention, folks. If you've already wetted your filter paper for your control at least, I want you guys to come up right here. Make sure, folks, that you're all paying attention because you don't want to delay them thereafter. Everyone can come in a bit closer. I want to show it here so that you're able to see the next steps. Everyone paying attention? You all ready? Okay. So you'll notice some of them are at the top of the lid of your tube, some are at the bottom. What do you do? You have two options. You can take them out. Sometimes you may have to get them partially. You have to get them. I'm not doing love, so and you guys eventually will do the following. And you can press it down, because as you put in a bit of force, they'll essentially be reactive, not too sensitive. So let's say I did this. That's your option. You have the other options. They're all just standing there. They're essentially structureless. I don't recommend doing this unless you find it very difficult and broken to me because essentially one of this is poking better. So you can do it like how it is right now. They all just start at the middle. That's your goal. So what you would do thereafter is something like that. Like all of them. No fun. I don't want you guys to get scared. These are organisms. They're not harmful. They don't bite. Some of them are. Bear in mind, though, folks. Some of them will be. So make sure all of them are out. Should be about 10 different sizes, but that's okay. Could make note of that for. But the goal is to take them all out, have them all in the center. So you can use your gloves, or I'm not like too concerned. You can use this glove as well. Gloves will make it easier so you can pick up more than one. And maybe move, that's okay. But the goal is getting them all in here and starting off with the center as soon as possible. If you're having difficulty with this for the control, let me know. I can help you. But you're going to be recording these results every minute from the time that you begin in your table do this. Let's go on to the day to close the edges. If you do the two and a half minutes right now, there's some definitely. You should be able to go. You find there almost stuff. Also do the I'm a little bit nice, but this is the last exercise on the Torah, so this year I officially on YouTube time. You get a time that you don't consider. Now you're ready. Now, have a look. Look at the ball. You don't want them to be obviously injured, so let me know what happened. Now, this is the control of this group. Now, once they're ready, and I won't do it now, but when you guys are ready, what you guys are going to do is essentially have a timer. Every person needs a timer, every set of pairs, because you guys, and this is why I wanted you guys to work as pairs to separate out the control of one string, the experimental of another. And that way, once all of 10 are in this middle well, when you are ready, when you guys are ready and you have your timer on, start with 0 and essentially stay in your table. It is page 71. Page 71 is where you're going to keep track of all of the different time points and where they're located. Your middle well is the one, the central well is the first column, following the time points. So just see that, like, depending on the number that you have, count them up and then put that at the very top. You need every member of your group to pay attention because you guys will be doing this in synchrony. Instead of you will be focusing on the one or the other, just make sure that you're paying attention and filling them up in the right table. This is for the experimental trial on page 71, and page 69 is for the control. So just bear that in mind. When your group is ready, and you remember adding in the filter paper for your control, you wetted them up with water, and for your experimental, only the middle of water, and 4 other wells with your other substrates on either side. You are going to thereafter placing the isopods into the middle well, close them into the center, count up the number, place them into your table, and then have a timer to start off the time. And every minute for the next 15 minutes, you want to record the data of where they move. So, for example, in your tables, you'll see the other columns, they'll show locations. Well 1, well 2, 3, and 4. You want to be able to take note of every minute where they're moving to. If you find the same to that same given region, continue writing down 2. But if you see a change from like 2 to 3 to 4, you want to be able to take those notes and essentially number or place those values accordingly to the wells that you find like in every minute. The next 15 minutes. Is that clear, folks? Is that okay? Any questions? Yeah.
What is the substrates on iron?
So great question. So folks, you have once again you have a control tray and it's an experimental one. Only for your experimental is where you're dealing with the ones in the red buckets. You could use the ones that are right in front of you, and that may make it easier. But if you wanted to use another, let me know. The idea is that use the ones that are in front of you, blot essentially the filter paper gently, slowly across the surface of the filter paper for your experimental trays, for only the ones at the edges. For the one in the middle of your experimental tray, for the one in the control, all of those will be just with water. You guys should all have enough water. Let me know if you don't, but don't overdo it once again. Filter paper before you begin should be not overly moist, but just sufficient for them to stay in fact and present in the trays themselves. Is that answer your
Well, right in front of us.
So only the experimental ones.
Right. Yeah.
All 4. So there's 4 different substrates for 4 edges. The middle well will be just with water, serving like a control again for your experimental tray. But only the control tray will have, or except the control tray will have all 5 wells covered with water on the filter paper. Any more questions? Any more questions? Look, people get on with it. I guess just that. What's the thing? What are they expecting? What's the experiment of this? Yeah, no, but you got to decide it now. Yeah. Oh, it's good. Anyone having any concerns? Remember, it's either in the water. Keep rolling about. Some of them may be that they're not alive. And we should be alive. In the middle well as soon as you can, and then just thereafter isolate, place in the sowbugs. You can even invert it and place it onto the affected dish like that. That is that all. Yes, you want to wait 5 minutes for them to acclimate before you count the technique.
Remember, waiting for 5 minutes for them to acclimate surface before you make surface. Please also make folks, you're not being harsh to bugs. Don't take out their lives. Don't like really like, try not to damage them. Yeah. Well, no. So that may make it easier for you to accordingly maintain the environment for the well. Are we paying attention? When your group is waiting, both let's pay attention. As you wait for the 5 minutes, and by the way, as an update, are many of you waiting in the 5-minute incubation for them to acclimate to the environment? Make sure to place back the glass cover so that way they can accurately acclimate and actually maintain moisture in the environment. Sowbugs prefer most environments. So keep that in mind. Yeah. Folks, so just bear in mind, once again, these sowbugs prefer like specific conditions. So just bear that in mind that whenever you are, actually, I'll ask now, what step are you guys on right now? I mean, 3 minutes for acclimating? The experiment? 15 or 3? You want to do 15 minutes? Oh, you're on the 3rd minute. I see. So, did you guys? Okay. So, folks, please make sure you place back for the entire time. This glass needs to be at the top because it maintains the moisture. So that way it doesn't, the filter paper itself doesn't evaporate with the liquid that you place. Folks, please pay attention that you're maintaining the environment for these sowbugs so they don't just evaporate, the liquid that evaporates from the filter paper, and you're just keeping track of timing. Remember, it's okay to go 15 minutes all the way through. Don't pause your timer if you can, so that way you can just try to record the values as you see them. But once again, have someone in your group to pay attention with the time and another member in your pair to record the values beforehand. But if you need a pause for like 10 milliseconds, that's okay. Just bear in mind that you are paying attention. For page 71, for those that do the experimental grade, you have the different substrates. Write them down on the top axis where you have the header available. So that way you can denote the different substrates that you're using. How are you guys? I hope it's going well, actually. I want to ask, how's everyone doing thus far? Are you guys incubating at 5 minutes or already done? The five. Perfect. And how about you guys? Are you guys 5 minutes in, or like did you guys already let them acclimate or no? Well, how many of you are doing the experiment as we speak, like 15 minutes in? Okay, okay. Uh, Dorlin, are you guys also doing it? The time points? Perfect. Okay, this is good. Keep it up, you guys. Keep it up. Yes, so I'm going to talk about that now. If you guys are done, or once you're done, folks, bear in mind that you're paying attention to your timer, and as well as I speak, very important that you guys are paying attention. That you need to understand. You guys have to place back the sowbugs back into that vial, and you could use the spoons to collect more than one. Before you do that, you need to actually use that paper towel, right? Reusing, recycle and reuse, recycle, reuse and reuse, reuse, recycle, reuse, something like this. So essentially, you guys need to do the following. Place the sowbugs here. Folks, are we paying attention? I know some of you may be looking at the timer, but I need to make sure you guys are paying attention. Please bear that in mind. I am able to know who is paying attention and who is not, so just bear that in mind, folks. When you guys are done with 15-minute incubation, while someone keeps an eye on time, place your cell bugs here, and then let them dry off their appendages with the water or substrates they're on, and then place them back with a spoon into, or even literally you can use this to pour them all the way down into into the tray or into the vial. And That's it. Let's go back to class. That always has to be there. Anyone else, by the way, please make sure that the environment is maintained. You guys can dry them out for about 3 minutes. Yeah. That way, because the beach and contacts are resting with surface at points for 15 minutes or for some time, you want to turn the back over from the center. And folks, I actually have this on it essentially this factor constantly. Okay, so I'm sorry. Yeah, I wish I could go. Okay, 50. One. Two. You guys can record the meet volume if you haven't done so already. I was thinking, if you wanted to directly mail make a copy. So if any of you need my contact,
ask you a question about
Yeah.
Yeah, and I also have a question. You know, I send us some emails and everything, but like you don't reply to them or that's not email address. It gives us an organization that I don't use to give the Excel sheet. I looked at that, someone else said that. It was I couldn't find you. Yeah, the Excel sheet. You know the data for it's owned by someone else. So I thought it was you, so I thought you would figure out that organization. Couldn't find you. Yeah.
So what you can do is check my friends.
I did. I did. It's,
Yeah. I think it's good.
okay. I couldn't find you.
Was everything okay? Or?
Yeah, no, everything was okay. It was just, I submitted my medical as a great family guy, so I submitted at the hospital. I had questions, but I did it there. So yeah, no, he died, but it's okay. So yeah, I just wanted to let you know, but like I couldn't find your email or anything. So yeah.
Thanks for doing that. I will say though, how long?
Oh, my great grandfather.
No, I mean the time.
Oh, 3 minutes.
Like I was about to say, there's no even this for me. Like, no, we're not going to do this. If you see it appear like that for whatever reason, just let me know. And let me.
Thank you.
No problem. Before you leave though.
Yeah, that's right.
Let's see. So you guys have already done this. Yes. And this is a reminder, folks. Please make sure, because each of you are doing different trays, control and experimental. Bear in mind that with the tables that you've drawn on pages 69 or 71, the control or experimental results, bear in mind that you calculate the mean and share with each other the data that you have. So that way everyone is able to complete the lab 4 assignment. That's lab 5 assignment, which is exercise 4 for your lab. If you guys have to leave, it's okay. But generally speaking, these are things that you guys need to know. So if you can stay to, if you guys have to leave, it's okay. Just bear in mind that you know how to use chi-square time. You guys know? Yeah, both of you. Okay, and then record as well the p-values. Share everything that's pretty much required in the lab 5 assignment. Yeah, pretty much what you see in the manual, but more details are described in the assignment itself. Pretty much what we talked about today. Do you guys have any questions? I imagine it's fun. Let me just see, in case you're missing anything. Wash your hands accordingly. Let me just see. Can I see your sowbugs? Can I see where they are?
The way you said perfect. I don't need to say we have, and so folks, if you guys haven't done so, take photos of each other's results for your lab 4, lab 5, full thought assignment. You get it all. Yeah. Take care, you guys. Filter paper. Yeah. I don't think we need. So I didn't say this, but keep in mind that there are other slides that you may find beneficial. So when your groups are done officially and you've joined the solvers for about 3 minutes and then eventually place them back into the file, let me know and I can go further with additional pointers for you guys before we end the second class. Yes. How many, is it okay if I move further actually, for those that are able to keep track with the slides? Let me just go briefly. We already talked about your independent variable, briefly about the condition, the environment that you're situating the sowbugs in, the Porcelio, SPP, erythropods or isopods. They're also shown as terrestrial isopods. So even though they live on land, they have external gills. Folks, let's pay attention. Pay attention, folks. So we have 3 different sources, right, of independent variables. And your goal as a dependent variable is to see how much they, or in which locations or wells do they migrate towards, right? So think about essentially the number, and that's what you're depicting as on your either your y-axis or as your dependent variable. And that depicts your response, essentially, the number of sowbugs in a specific well or section. Now, which variables do you need to control for? This is such a great question. I want you guys to think about this and maybe give some examples for control variables. So think about what I've talked about earlier today. Think about even the environment. Think about the setting, the lights, the humidity. Think about how much of essentially filter paper you use. Think about the substrates themselves. For example, the control in the middle well, you had it for both the sorry, I don't want you guys also to bear in mind. Please keep the lab as organized as possible before you leave. But think about all these things that I just touched upon. Think about like your FLR, right? Humidity, air exposure, water, etc. were controlled. These are things that you want to also keep in mind and take into account. We also did this. We had 4 different choices for the experimental tray, and all of them were constant with water, with the filter paper for your control tray. Once again, be gentle. Be gentle also when you're bringing them back into the vial, into the test tube with the sowbugs. So that way you'll be able to rest assured that they are safe and you haven't damaged them in any way. Let me just get an update. Everyone's okay? Any questions from previous slides or anything at all? Everyone's cleaned up thus far? And so we're going to talk in due time, once again about something very important. But in the meantime, we're actually and I want to make sure that you guys are well prepared for that. So this is very important. As you know, hopefully, that you were dealing with the chi-square test for stats. So essentially, from the values that you've derived thus far, from the different values across the different wells, you're going to be looking at your last time point at 15 minutes. What were the number of sowbugs, the sowbugs, the number of sowbugs in each of those wells for your control and your experimental treatment or trade? And that's where the chi-square test comes in, because you're trying to see if there's an association to particular wells based on the values that you have. So bear in mind that you have all of the wells with their identifiable categories. For example, we have for the chemical treatment, or sorry, for the food substrate, we have fat, protein, fruit juice, starch in that specific order. Once again, you need that in your tables at the top of page 71. But you need to make sure, folks, you get all of this data and you deliver it to me through your post-lab assignment. So what I mean by that is that you'll include the chi-square results, which you'll see. Actually, let's go down into your manual. You'll see in page 72. So once you've collected your data, pages in page 70 and 72, you'll be able to depict your chi-square test, and you need to be able to essentially record based on the categories. You need to know the values of the wells at the last time point, but also of the names of those categories. So I set them for the group on my right, but you need to have them specifically denoted. And thereafter, your chi-square test will show a comparison between your observed versus expected results. Your null hypothesis assumes that there's no significant difference between what you observe and what you expect. Folks, please bear in mind you're paying attention because you need to have this for your post lab. You have, for your control, ideally 10 different sowbugs in 5 different wells. For control, you would assume that it's equally distributed such that there isn't an effect, right, between your observed and expected results. And what that means is for your expected, 10 divided by 5, 10 sowbugs, it's 10 bugs, divided by 5 wells, you should expect to see 2 sowbugs per well, right? And that would be assumed for your control as well. If you don't, this is something that you want to highlight for your post lab as well. But essentially, you want to have an even distribution. Compare that with what you observed from your experimental data, from the pair or set of members of your group that have reported this data. And thereafter, include this with your mean values in table 5-3 for your control and table 5-5 for your experimental trial or trade. And bear in mind that you're doing the calculations of your observed, what you've seen from your tables, 7.1 and 7.2, or sorry, 5-2 and 5-3, 5-4, pages 69 and 71. Do you record those data points? You're able to thereafter determine the chi-squared value. That is your value that is calculated, and you're going to compare that with what would have been expected. If your critical value, based on the table that you see in your manual, is essentially, let's go back just so you guys understand this, very important for stats, and I think it's also here. You can see it if we go down. Yes, with your p-values, similar to what you've done for your FLR, if it's less than 0.05 or equal to, you reject the null hypothesis and assume that there is a difference between your observed and expected data. Or you reject your null hypothesis and you appear to support your alternative or research hypothesis. And then remember the degrees of freedom. You had 5 different wells, -1, that's 4 degrees of freedom. And you use this formula to calculate for all choices the chi-squared calculated value. And thereafter, using the critical value that you use from this table here at 0.05, that's 9.488. You need to compare that value with what you would have gotten from the formula here for all choices. If you're calculated, and so just so you guys understand that, that's for your control and experimental trial. If you find that your experimental or control is greater than 9.488, then that's where you depict what we've talked about before, that it rejects the null hypothesis, and likely you'll also have a p-value less than or equal to 0.05. The converse is if you have a critical value that is greater than you calculated, or a p-value that is greater than 0.05, and thereafter you cannot reject, or you fail to reject the null hypothesis. And thereby, you would assume that for your condition, right, of food, chemicals, or of sugars, that thereafter, cell bugs do not appear to have a significant response to to your specific condition. It could be chemicals, sugars, or food, etc. And pretty much for the second last slide, because I believe all of you have cleaned up pretty good. Great job, well done, everyone. Remember to include this data. Think about what I've talked about thus far and be able to answer questions. Any final questions? Sorry. Anyone? Any questions? So remember, this assignment's due 24 hours from 1 p.m. today. So make sure you do it as soon as you can. Try to record the data, do it independently, and do it to the best of your ability. And leave the best with the rest. And so once again, grateful to have you guys. This is your second last lab, and I hope you've come to appreciate the different organisms we've examined thus far, and hopefully are able to see how they connect to your final lab in 2 weeks' time. Take care, everyone. Have an amazing week, and let me know if you have any questions. Take care. Take care. Yes.
So both you control and experimenting. Okay.
No problem. No problem. You had 10 samples, so do you know how to do the calculation? Do you know how to get the expected value?
Numbers of which is 10 divided by category, which is 5, just 2.
2, yes. So hopefully that helps you guys out for your expected value. Expect that to be 2 if you had 10 starting sowbugs. If you had 9 or 11, then you just divide by 5 accordingly. But you include that with your expected and replace and compare, those values with what you've observed. Calculate the chi-squared values from the slide, from your manual, from your results, and see how that fits well with the t-test for your p-values as well. Thereafter, discuss what we've talked about, the questions, state your research hypothesis, null and alternative, and predictions for each trial for your control and experimental. With that, there's additional questions that you'll see from your lab manual that will be part of your assignment. Have a look there. All the best, everyone. Take care. Have a great day.
Is it like, I don't know if it is great. Actually, fine.
It depends.
In what way? You know, like, I want to be research on it.
Okay, that's a good idea. So, you can lay yourself looking into
Actually, let me ask, what type of theme topics are you?
Anything like any
Like in science, like in biology, or like in physics, or
often it's like a wide range. So I have one thing that's like anthropology, one thing that's like biology, like three things that are biology, everything. Yeah, so it's like anything like science related, that's what that.
you mean like fossil research? When I think about anthropology, I mean, but I know there's other like it just came to mind because I actually there's a member that I knew or I spoke to or like attended one of his talks. He was actually doing anthropology research at a conference I attended in the US, and he was talking about it was actually at Harvard Medical School. So he was talking about his presentation about in Boston or in the Bronx area. So this is in New York. There is like uh, black ancestors that essentially have fossil records that are being either missing or outdated. And so he was actually trying to explore, trying to look into the cemeteries and stuff, and like really showcase a lot of interesting things. He used biology as well because he was looking at their essentially uh, decomposition and the nutrients that they have. And there's things like that. They're maybe a bit niche, but you can look in like different like research articles or literature on that may compelling. Or from the lectures. I actually did a
That's why it's like I find it like I'll like take it from the lecture.
yeah.
Like there was this one that I did like orchids. Like they were
Yeah.
talking about like orchids and how they make bees that attracted them.
Okay, yeah.
Yeah, I did well on that.
Did you include that for yeah, I think I remember someone also talking about that.
Yeah, the one that was, it's like really long. I think it's like so far it's like 27 pages. But I'm doing right now it's like language and like okay, so it's like how